anxious when we need to know what do we found since last time on the discourse, and apparently get plenty of new things to teach us. I'm very proud and happy to get here one more time here. Eugene.
First of all, thank you very much, Didier, for the lovely introduction. It's always a great pleasure and an honor to speak here. Beyond that, what I'm going to discuss here is in a certain sense, a tribute to Didier. As you can see on this word rhizome that I put in the title of my talk, which you will not have on the poster or on the announcement, but I thought that this really would be the best term to introduce succinctly what I want to tell you. I want to talk to you about the diversity of the sources in the evolution of eukaryotes. Now, if I understand correctly, most of the audience here includes microbiologists. We work on bacteria and viruses, and you might wonder why this is relevant at all. But I hope we should see during this talk why this is very directly relevant. So basically, a eukaryotic cell is a very complex affair. It's a highly compartmentalized, elaborate ensemble of molecular machines that includes the nucleus, the chromosomes are located, a whole system of endomembranes and the plasmic reticulum, the Golgi complex and all of that, as well as the mitochondria, the power stations of the eukaryotic cell. And it's no secret anymore, and no matter of dispute, that the mitochondria evolved from symbiotes, or more specifically from alpha-proteobacterium and the symbiotes. It's interesting though to just see that. And this is why, this is the main reason I'm showing you this slide, not just to emphasize the complexity of the eukaryotic cell, but to visibly, graphically illustrate to you the relationship between the mitochondria and intracellular bacteria. In this case, it's the plant cell, which is really packed with, infested with rickettsia-like bacteria, small small intracellular bacteria. So we can sort of visibly imagine the path from, roughly speaking, from here to here, to the mitochondrial and the symbiosis. So, but all that is good. My trade generally, what I really do is comparative genetics. So for most of this talk, we will talk about genes and evolutionary relationship of genes. And speaking of that, in evolutionary terms, the eukaryotic genes is a mix, is a chimera of archaeo genes, bacterial genes, ancestral genes of origin that we don't know, and some eukaryotic-specific genes. Right now, I, however, want to concentrate on the combination of bacterial genes of the parent bacterial origin and genes of the parent archaeo origin in the eukaryotic genomes. And this comes from the work of my long-term collaborator Bill Martin and his lab done several years ago. And you can see graphically here this group of genes of apparent archaeo origin right here, and this group of genes of apparent bacterial origin. So in the yeast genome, to be specific, about 70% of the genes that do have any homologs at all in prokaryotes appear to be of bacterial origin, and the rest about 30%, appears to be of archaeal origin. I hope everyone in the audience knows what archaeal bacteria are. They're the two distinct kinds of prokaryotic organisms, formerly called archaeobacteria and eubacteria, but we now understand that the distinctions between is such that it's best really to speak of archaea and bacteria separately. So it's this kind of chimera, the eukaryotic gene set, and moreover, there is a very clear functional split between genes of apparent archaeal origin that are mostly involved in informational transfer processes, such as replication of translation, and genes of apparent bacterial origin, which are mostly operational genes, metabolic enzymes, and so on. It's a remarkable observation that is at some level well known, but I think still underappreciated. So genetically, eukaryotes are archaeobacterial hybrids. And this, I think, immediately begs the question: how did this chimera emerge in evolution? And how does this chimeric organization of eukaryotic genomes relate to the key innovations of the eukaryotic cell? So, the evolution of eukaryotes. So some 20 years ago, 15 years ago, even 10 years ago, apparently the prevailing view of the evolution of eukaryotes could be formulated in terms of the so-called archizo hypothesis, which is a very logical scenario of evolution. This scenario maintained that at the early stages of evolution, there was a proto-eukaryote, which was much like modern eukaryotic cells, with a nucleus, with a cytoskeleton, with a system of endometrium, just without the mitochondria. That also has the phagocytosis ability. And through that ability, it engulfed the mitochondrial ancestor. Now, supporting that scheme were the so-called archies, anaerobic eukaryotes that have no typical mitochondria. And there are many of them, such as, for instance, diplomonads, such as well-known parasites are there, most of them. Diplomonads, such as giardia, parabassalids, such as trichomonas, microsporidia, and a few other more obscure proteins that didn't seem to have mitochondria. And when you build the phylogenetic trees using the sequences of RNA or using the sequences of highly conserved proteins, such as RNA polymerase subunits, these seem to sit here at the root of the eukaryotic tree, very compatible with their status as primary and mitochondrial eukaryotes, archizones. So all this was quite well organized and neat, except later on there emerged a problem. And more than that, it turned out that there are no true mitochondrial eukaryotes, because all these so-called archizones actually possess mitochondrial genes in nuclear genomes, and in addition to that, they have degenerate derivatives of the mitochondria, such as the hydrogenosomes or the mitosomes. These organelles are really not quite very typical mitochondria. In particular, importantly, they do not have genomes, they do not have chromosomes, they do not have their own information processing system, but it turns out that many proteins that go to these organelles are actually mitochondrial genes coming from the nucleus genome. So basically, all these organisms are not archizones at all. What we are facing here is the situation where this endosymbiosis of the mitochondria is seen in all extant eukaryotes, and by extrapolation, was already there in the last common ancestor of all the existing eukaryotes. So this really changes the way of thinking about the origin of eukaryotes. So this somewhat complicated scheme, which I will not discuss in every detail, comes from a really nice review published in the Nature magazine some years ago by Martin Emily and the already mentioned Bill Martin. And this review classifies all the scenarios of the origin of eukaryotes into the rhizome hypothesis that postulate the existence of primary mitochondrial eukaryote, it was the host of the mitochondrial and the symbiont, but which we do not observe among any existing eukaryotes, and the symbiotic hypothesis, according to which the symbiosis actually was a primary event of the trigger of the evolution of all the innovation, of all the complexity that emerged in the eukaryotic cells. So, given the absence of primary mitochondrial and the primary mitochondrial eukaryotes, these hypotheses now should be considered seriously. And they may vary in terms of the nature of the specific host, the nature of the specific endosymbiont, the mechanism of endosymbiosis. All these issues are open to debate, but the primary divide lies here, with the endosymbiosis after eukaryogenesis, so to speak, and endosymbiosis triggering eukaryogenesis. Right. So under the, here I want to become a little bit more general for a couple of slides. The endosymbiotic hypothesis, if you think of the endosymbiotic scenario of the origin of eukaryotes, if you think of it, is actually quite revolutionary with respect to our whole understanding of biology. Seriously. Because what I showed here is a very simplistic representation of the classic tree growing. Issues floats here, but they're mine, or the tree of life, that goes to the classic studies of carbon wars, and this particular representation comes from a more recent paper by Norman Pace, second quote. Comparisons of ribosomal RNA sequences reveal a three-domain tree of life, rendering the term prokaryote obsolete. Well, we don't really care that much about the terms, but the issue is that looking at the structure of the tree, it's easy to understand what Norman meant, because eukaryotes here cluster with archaea. This is very reliable. This can be reproduced by phylogenetic analysis of ribosomal RNAs, ribosomal proteins, translation factors, RNA polymerase subunits, and some other proteins primarily from the information processing system. So that is supposed to be the tree. However, the endosymbiotic scenario really changes this. Instead of the tree of life, you must think about something like this. Namely, the genomic fusion of bacteria and archaea, giving rise to what I call the first eukaryotic common ancestor. And of course, on top of this, in these early stages of evolution, there was apparently a whole lot of horizontal gene transfer. We will have the opportunity to discuss this. So, three domain under this symbiotic scenario, the three-domain tree of life. The three-domain tree is no tree of life at all. Actually, even more important, there is no such thing as a tree of life. Because in mathematical terms, a tree is a bifurcating acyclic, directed acyclic graph. And this is a graph with a cycle. So this is not a tree at all. Which is why I call this scenario revolutionary. So over the next 15 or 20 minutes, we'll examine a variety of evidence that I hope will convince you that I'm deliberately dropping the suspense. I hope will convince you that this endosymbiotic scenario of the origin of eukaryotes is a better scenario than the is a better scheme than the Archizon hypothesis. And we'll get some details of that. So, the first thing that I want to address here is the role and impact of introns in eukaryogenesis. And the question that I want to ask is, was it possible for eukaryotes to emerge without actually having these genes in pieces? Of course, I hope that everyone is on board. The majority of, the great majority of genes in our genomes are interrupted by non-coding. I'm talking about protein-coding genes. The great majority of protein-coding genes are interrupted by non-coding sequences called introns. And actually, all eukaryotes possess these complex features. They have a nucleus with advanced nuclear prokaryotes. Beautiful, highly complex structure. They also have mitochondria. And they all also have at least a few introns. And they also have a highly complex molecular machine for excising those introns and joining together exons, the common parts, called the splices. And of course, they have other complex features, such as the cytoskeleton. So, and all eukaryotic cells, and accordingly, the reconstructed last common ancestor of all eukaryotes, come equipped with all of these complex features. So, naturally one starts to think, could there be a causal connection between the emergence of these various complex features? Mitochondria, introns, nucleus, and possibly more. So, continuing on this subject of introns, I will provide a quick perspective from the point of view of population genetics. And I will try to show you that phylogenomic study of introns can tell us something important and possibly quite a bit about the origin and early stages of the evolution of eukaryotes. So this figure comes from a very recent, latest reconstruction of the evolution of the structure of the eukaryotic genes, the gain and loss of introns that we published last year with my colleagues. And to make a long thing short, what I want to point out is that maximum likelihood reconstruction of intron gain loss process during the evolution of eukaryotes by analysis of 100 representative genomes reveals extremely I think in a counterintuitive or unexpected manner, reveals highly intron-rich ancestor. Simply put, the last universal common ancestor of eukaryotes, according to this reconstruction, that has quite strong statistical evidence supporting it, had an intron density very similar to that in our human genome right now. Moreover, many lineages, if you trace the lineage from last common ancestor to humans, right here, you will have no intron poor stage the genomes the genes remain intron-rich, densely packed with introns all the time. Not all the lineages are like that. Many have lost introns many of the introns on a variety of occasions. But basically for the ancestral forms, we confidently reconstruct the intron-rich stage. So it just shows you the confidence intervals here, and it's interesting to see, in particular, the rather narrow, highly reliable confidence interval here from the last eukaryotic common ancestor, and also bring your attention to this panel. It shows that at the base of the animal evolution, there was even addition of intron, apparently. There was even addition of introns and very intron-rich stage. So these reconstructions suggest something rather incredible about the genome of the last eukaryotic common ancestor. So in order to understand this, we need to recall, tell you, that lysosomal introns, the introns interrupting the eukaryotic protein-coding genes, come from so-called group 2 self-splicing introns that are found, that are particular type of mobile genetic elements that are found in bacteria, some archaea, and mitochondria of proteins, fungi, and plants. Structural comparisons leave no doubt whatsoever that lysosomal introns have evolved from these self-splicing introns, particular type of genetic elements. And these elements have a very uniform size, about 2.5 kilobase. So at the invasion time, and from the evolutionary reconstruction that I showed you before, we are inclined to believe that this was quite a rapid invasion. So at the invasion time, the average size of introns was like this, between 2.5 and 3 kb. So the introns have dramatically shrank in all modern eukaryotes, except, interestingly enough, for our own lineage, we do have very large introns. So in that respect, apparently the genome of the last common ancestor of eukaryotes was quite more wasteful than the modern eukaryotic genomes. And more dramatically, it's very easy to estimate that genome of the last common ancestor of eukaryotes actually consisted mostly of intronic DNA, by about 80%. The greatest fraction of genome occupied by introns these days is seen in plants, and it's a bit less than 50%. Now, the genome of the last eukaryotic common ancestor most likely actually consisted primarily of introns. Now, I will not walk you through this estimate. We don't really quite have the time to do so. I will just say that I'm taking a page from this important book of Michael Lynch on the non-adoptive origin of the genomic complexity. A very, very important theory that I think is largely true. And using the formalism that Michael Lynch introduces in the book and other publications, what we can estimate is that at the origin of eukaryotes, there was an incredible population bottleneck, which was the conditions, the necessary condition for the fixation of all these introns. The effective population size being very tiny, probably less than 10,000, which indeed is a situation that allows a whole lot of genetic drift and the fixation of various kinds of innovation through the aforementioned invasion of introns, through duplication and other processes. So at this point I'll switch gears somewhat and discuss in a more biologically specific manner the nature and origin of the key innovations in the process of eukaryogenesis. Of course, if you think about the very word eukaryotes, well, in Greek that means those that have a true nucleus, the real nucleus. So it's natural to ask the question, why would you like to have a nucleus? So the raison d'être for having the nucleus is actually very unclear if you think of it. Because the existence of the nucleus dramatically complicates the function of the cell by suppressing, eliminating the highly efficient coupling between transcription and translation that is typical of bacteria and archaea. And on top of that, not only disrupts the emergence of the nucleus, not only disrupts this coupling that requires a complex export-import machinery for mRNAs and proteins. So one would strongly suspect that there will be a powerful driving force behind the evolution of such a complex machine as the eukaryotic nucleus. So the hypothesis that we proposed with Bill Martin a few years ago is this, that the nucleus actually evolved as a defense system against massive invasion of introns into protein-coding genes, because think of this. Without compartmentalization to separate intron-containing transcripts from the ribosome, and to allow only mature, correctly processed mRNAs to be translated, there would be tons of aberrant proteins accumulating in the cell, probably actually killing the cell, especially because splicing, for all we know, splicing is a much slower, by about 2 orders of magnitude, slower process than the process of translation. So as we have seen, there was an early invasion of huge numbers of introns into the genes of the emerging eukaryote, creating something like a nucleus, a defense system that isolates the primary transcript and allows them to be processed, to be properly spliced, is the only way to survive that situation. Why suddenly an intron invasion? Well, we don't know that, but the speculation that I think is very plausible is that this is what happened in the wake of the mitochondrial endosymbiosis. Namely, the symbionts group 2 introns, cell-splicing introns, went on a rampage within the chimeric cell, probably through a combination of weak purified selection caused by the small effective population size of the chimera and the absence of specific control mechanisms in the host archaea. So the schematic scenario which we proposed with Bill Martin looks like this. The evolution of eukaryotes starts with 2 prokaryotes, an archaean host and the alpha proteobacterial invader, the ancestor of the mitochondrial endosymbiosis. The invasion creates the unidirectional, creates a unidirectional flow of genes and introns, of DNA in general, from the symbiont cells due to the propagation of multiple copies of the symbiont, multiple symbiotic cells, and lysis of the symbiont in the chimeric cell. This is completely a non-adaptive process. It's basically an attack of parasites on on the host genome. Now what follows is the adaptation to survive the intron invasion. So this invasion apparently led to an extremely thin, slender population bottleneck. Those that survived, those that adapted to survive the intron invasion. Above all, by evolving the nucleus and the splices. More generally, I could actually talk about that for a long time about various molecular details and logic here, but to cut the whole thing short, I think I can propose a single causation chain that explains a variety of adaptations in the a variety of complex features of the eukaryotic cells that are common to all eukaryotic cells by adaptation to the survival of that invasion of genomic parasites that became introns in the eukaryotes. So basically we have spoken mostly about the origin of the nucleus, the results of the propagation of the membranes of the endocyte itself, and these membranes yield the nucleus, but also the other aspects, the splices of the linear chromosomes, the nonsense-mediated decay, the ubiquitous signaling system. All of these are different echelons of defense against the invading genomic parasites, according to this hypothesis. Okay, so that much about the eukaryogenesis. I want to somewhat change the direction of the discussion now and try to justify the word rhizome in the title of my talk. And in doing so, I'll introduce this term, the eukaryote, which is a term of my own invention, which is a very easy invention, because there is innumerable number of ohms nowadays around. One can call anything an ohm, so I call it eukaryote, which, by the logic, is the entirety of eukaryotes or of genes that are, in a sense, essential, that are signatures of the eukaryotic cellular organization. And it's easy to list the cell structures that comprise the eukaryote, the nuclear complex, the histone basics, chromatin, the splices on the RNA system, and so on, and so forth. So all these systems are traceable back to the last eukaryotic common cell in a complex, nearly modern state. So we had this strange impression that all these complex systems came out all at once, like basically Aphrodite from the foam in the Mediterranean, in the emerging eukaryotic cell. So, of course, the tree-centric thinking in evolutionary biology that I tried to partially denounce in the very beginning of my talk is quite deeply rooted in evolutionary biology. So, people tried rather hard over the last 20 plus years to trace the specific origin of eukaryotes with regard to archaea. And this was done first by exclusively analyzing the ribosomal RNA sequences, because there was not, actually started to be more precise, it started with the ribosomal structure even, but then quickly went to the ribosomal RNA sequences, as in the famous 1988 paper of Jim Lake. But then people started using more and more highly conserved genes because the gene was become available and it was an opportunity. And basically, as I said, under this paradigm, the archaeo ancestor of eukaryotes is really elusive, depending on the techniques used, on the sets and on the set of genes used, or the so-called, and on the, probably also on the prejudices of the researchers who do this, the so-called origin of eukaryotes is basically all over the phylogenetic tree of archaea. The already mentioned paper of Lake, followed by subsequent studies from his lab, map the eukaryotic ancestors to the Koonin, others mapped to so-called Tau archaeota, the new phylum that was previously known as mesophilic Koonin archaeota, yet others mapped to the Uri archaeota, and so on and so forth. I'll credit our own work of a few years ago by at least putting out the notion that different genes, different conserved genes of eukaryotes show phylogenetic affinity with genes from different groups of the archaea. The majority actually branch of here deep in the archaeo tree, but there are substantial minorities that seem to come from different archaeophytes. In any case, all possible candidates have been proposed over the time. Now, actually things in that respect, in terms of the topology of the tree for universal prokaryotes, seems to be stabilizing. It seems to be stabilizing on the affinity of eukaryotes with what has been nicely called by Dice Etema, has been nicely called the tech superphylum of archaea, which includes the Koonin archaeota, Tau archaeota, Core archaeota, and some more obscure archaea. So this seems to be settled. It is the origin of this component of the eukaryotic cells. But we observe something that is, I think, more interesting and potentially more important than the actual interception. This comes from a from a still unpublished phylogenomic analysis of ribosomal proteins that we did in the lab. And what we find here, that among the 50 or so ribosomal proteins, there are 12, good fraction, that are present universally in eukaryotes, but lost in one or other archaeal lineage. Basically, all archaeal ribosomal proteins are represented in eukaryotes. But in individual archaeal lineages, many have been lost. Some here, others here, and so on, and so forth. So you see this ancestral complexity coming out in eukaryotes, but being streamlined in different archaeal lineages. And I will say that this is the pattern that is recurrently seen through analysis of great variety of conserved proteins. So in the interest of time, because I have another subject to discuss that I find quite exciting, I will go quickly for the next few slides. But it's the same pattern. Basically take this subunit of RNA-dependent, DNA-dependent RNA polymerase. This is shown by all eukaryotes, Koonin archaeota, and Core archaeota, not the rest of the archaea. Same pattern here in another RNA polymerase subunit. Shown by eukaryotes, Koonin archaeota, Tau archaeota, not the rest of the archaea. Now, some patterns are actually quite complex. For instance, when we analyzed the evolutionary history of all eukaryotic DNA polymerases, the enzymes responsible for DNA replication, we saw a very complex picture with two distinct forms of DNA polymerase inherited from different groups of archaea and possibly yet further inherited from bacteria. So basically what happens is that the highly complex organization of the eukaryotic DNA polymerases involve contributions from different genes of archaea and possibly even bacteria that are found in different groups. And very interestingly, this combination of elements of different origins even occurs within one protein, such that, let's say, one domain or polymerase domain comes from CRAN archaeota, whereas this domain, the zinc finger domain, comes from the uri-archaeo DNA polymerase. So there is a veritable mosaic of components coming apparently from different groups of archaea in the eukaryotic replication machine. Now, it's particularly interesting to trace and discuss the history of the eukaryotic cytoskeleton and the cell division apparatus. Because this is one of the key complex features of the eukaryotic cell, but it's also important particularly important for understanding the origin of eukaryotes, because in mechanistic terms, these are the components of the cell that would be responsible for engulfment of the endosymbiont. And here what I would like to emphasize is the importance of genome sequencing, importance of sampling diverse genomes from the environment in particular. Because the more and more we sequence archaeal genomes, the more interesting things, the more components of that eukaryote we detect. In this particular case, it has been known for a while that archaea and bacteria have very distant homologs of actin, the key component of the eukaryotic cytoskeleton, which has become apparent. The increased representation of sequenced archaeal genome is that a certain assemblage of archaeal group, Thermopatalis or archaeota, and something called archaeum, to which we'll return in a few moments, actually have true actin homologs, or proteins that unequivocally, very clearly cluster in phylogenetic trees with eukaryotic actin. And actually you can just see that by the sequence similarity, that these proteins are most similar in terms of sequence to the eukaryotic actins rather than to any other proteins from prokaryotes. And lo and behold, we have done this prediction by phylogenetic analysis, and Settema and his collaborators confirmed this prediction experimentally by discovering what they call renactins. Very recently, just earlier this year, we realized that the same exact thing pertains to the tuberous, to the other key component of the eukaryotic cytoskeleton. A couple of very recently sequenced genomes of tau archaeota, previously known as mesophilic CRAN archaeota, contain proteins that are most similar, very clearly, to eukaryotic tuberous and very reliably group at the base of the eukaryotic tree in the subtree in the phylogenetic trees compatible with the idea that these are indeed the true ancestors of the eukaryotic tuberings. This is now being tested experimentally, but the message is very clear. The ancestors, very believable, reliable ancestors of the key component of eukaryotic, of quintessentially eukaryotic machinery, such as the cytoskeleton and the others, are seen in the various groups of archaea. We have a couple of years ago extended this to the more broader study of the cell division systems, and that brought up very interesting results. Namely, that there are at least three distinct types of cell division systems in the archaea. One is based on the tubulin-like protein FTSZ, which is sort of bacterial-like. The other two are very much eukaryotic-like. And this is called based on the so-called escort-3 complex and on the actins that I already mentioned to you on the previous slide. Many archaea have combinations of two of these systems, but none has all three. Evolutionary reconstruction suggests that the archaeal ancestor could have been more complex than any of the extant forms, with all three cell division machineries combined, as illustrated in this schematic, according to which both the ancestor of all currently known archaea and the ancestor of the eukaryotes, whatever is its exact relationship with those archaea, already had all this complexity with respect to cell division and membrane remodeling mechanisms. So basically what we suggest is that the archaeal ancestor of eukaryotes, which according to the symbiotic scenarios of both the host of the mitochondrial and the symbiote, might have been an organism with a primitive form of actin-based cytoskeleton that facilitated engulfment of bacteria and supporting the domestication of the future mitochondria. Well, this is not all the story. As I said, sampling of diverse genomes is enormously interesting and important. And about a year ago, this unusual archaeal genome, so-called Caldi archaeal subterraneum, that the Japanese lab isolated from deep into some kind of gold mine or something like that, it had been published. What I saw there surprised me at first to such an extent that I thought that this could not be true, or that this would be some sort of contamination. Namely, this genome encodes the prototype of the entire ubiquitin system. With the ubiquitin-like protein, in the same organ, with the ubiquitin-like protein, with the E2 ubiquitin-like subunit, E1 ubiquitin-like subunit, and the ring finger, the E3 ubiquitin-like subunit. Now, the examination of the genome sequence shows that this is really contiguous with other genes, so this is probably not a contamination. And then again, when we look carefully into genomes of certain particular bacteria, we see very similar operands. So, what I believe now is that this is not a contamination of these genes are really there. And then we make the phylogenetic trees, lo and behold, these proteins indeed cluster very clearly with the eukaryotes as opposed to prokaryotic distant homologs of the ubiquitous system proteins. And this, I will not go into this in detail, but this holds for each component where we have sufficient information for reliable phylogenetic analysis. Before I come to some conclusions on the origin of the eukaryote, I want to show you this, the available information and conclusions on the origin of the eukaryotic RNA interference system, because it brings another important aspect. I just show you this slide from a rather old review by Eric Sondheim to remind you of what the RNA interference system is. Basically, CHOP, or the double-stranded RNA by the nucleus or Nucleus. DICER, which is then incorporated to the so-called RISC complex, and that complex uses the small RNA as a guide to destroy the target homologous mRNA. Now, this is very well known. What is less well appreciated is the complex origin of the components of this RNA system. So DICER is a very complicated protein that contains several domains, in particular helicase domain, the double-stranded RNA binding domain, so fast domain, which is protein-protein interaction, and two RNAs, three domains, which are actually the effectors of the nucleus responsible for processing of RNA precursors. So this is very interesting, because when we do phylogenetic analysis, we very clearly see that the helicase domain of DICER has an archaeal and more specifically uri-archaeal origin. Here are the DICERs with the uri-archaeal homology, different kind of helicase. But archaea essentially even do not have RNase 3. RNase 3 is a typical bacterial enzyme. What this quintessential eukaryotic defense and regulation system is a very complex archaeobacterial chimera with different components having either archaeal or bacterial origin, and then other components apparently having a bacterial phage origin, in particular the RNA-dependent polymerase that is involved in the amplification of the small interfering RNAs. So basically, what we have to conclude here, in a nutshell, is that the eukaryote is truly chimeric. When we take a look at the modern prokaryotes that we are aware of, for which we have genomes and so on and so forth, there is a substantial bacterial contribution, and then there are contributions from different kinds of archaea apparently. When our colleagues made a rather sophisticated maximum likelihood reconstruction of the common ancestor of all the modern archaea, and we are now expanding the reconstruction, they came to the realization that most archaeal lineages are characterized by net loss of gene families. In other words, the main trend in the evolution of the archaea, which reappears convergently in different lineages, is streamline, is loss of genes. The common ancestors of the archaea, of the most archaeal groups and of all archaea, are reconstructed, shown schematically here, as large, more complex genomes than the majority of the modern forms. So putting it all together, I want to propose the following scenario of the eukaryotic origin. Very schematic. It's something which I call the ape scenario, the transient archaeoprogenitor of eukaryotes, which exchange genes with various bacteria and archaea. Basically, the idea here is that the host of the mitochondria and the symbiote was a very complex archaeoform. The variety of genes, a variety of features, and probably existed for a relatively short time on an evolutionary scale. But that time was sufficient to acquire the mitochondria and the symbiote and to trigger eukaryogenesis. There is the evolution of most of the archaeo lineage, primarily characterized by genomic strain variety. Well, basically, to recapitulate, what is the best scenario for eukaryogenesis? The rhizome scenario or the endosymbiotic scenario? Strictly speaking, we cannot distinguish. But there are two very important things to keep in mind. One is that we are unaware of any existing living archaeosaur. It's not impossible in principle to imagine that they have existed but have gone extinct. More importantly, however, with the archaeosaur scenario, we really have no clue as to the origin of the complexity of the molecular complexity of the eukaryotic cell, whereas the endosymbiotic scenario, there seems to be a single chain of causation for the major eukaryotic innovation. Moreover, it's interesting to consider that the engulfment of one prokaryote by another is not an impossibility. First of all, think of the cytoskeleton that is seen in some archaea, but also think of experimental evidence. Indeed, prokaryotes living inside other prokaryotic cells have been described. It's not a common occurrence, but it does exist. So basically, the idea is that we really should not be looking too hard for a single ancestor of eukaryotes among known archaeo lineages. Evolutionary reconstruction implies complex archaeo ancestors. The likely ancestor of eukaryotes in the hopper and the symbiote might have been a highly complex, perhaps transiently existing archaeo lineage. We can surmise that this is a boneless mesophile that probably possessed actin-based cytoskeleton that could facilitate engulfment of other cells. How can we understand the origin of the eukaryotes? I think it's very relevant to mention this here. Basically, extensively sampled archaeo genomes and search very carefully as we try for eukaryote elements comprehensively across the archaeo diversity. This is what we're trying to do right now. To finish that part of my talk, and I will try to complete the remaining part in 3 or 4 minutes, although there is much more to say. I'll just show you this. These are huge archaeo cells, mesophilic tauarchaeo cells. These are covered by bacterial ectosymbiotes. Now, these are not yet endosymbiotes, but it's a small leap of imagination to think that the possibility might occur for these cells to grow inside. Alright, so I practically have no time to talk, but I do have a different subject, and I will go through this subject extremely quickly. And my apologies for this. So that subject is the horizontal gene transfer from bacteria to Unikonts, which is a group I'll elaborate on this in a few seconds, but basically this is a group of eukaryotes that includes animals and fungi, and along with a few others. Basically, many people, I'm sure in this audience, recall, well, we have very young people here, but some do recall the first draft human genome paper back in 2001. Now, it was a pretty boring draft paper, but there was an interesting thing there. And that was a table that listed genes that possibly have been horizontally acquired by animals from bacteria. Now, that has been vehemently denied by many people. And the consensus has been more or less established that the appearance of horizontal gene transfer beyond genes from NSC in multicellular eukaryotes, most especially in animals, is explained away, can be satisfactorily explained away by multiple gene loss and artifacts of phylogenetic analysis. Now, all this is very real, and I think the list of the potential transfer genes in the 2001 Human Genome paper was not particularly reliable because we really underappreciated the gene loss at that time. That being said, a variety of anecdotes keep accumulating, strongly suggested the possibility of horizontal gene transfer from bacteria in some animals and the biological role for that transfer gene. There is a variety of such communications, some of which speak of multiple genes being transferred. And there was some more systematic effort, in particular the recent study of the acquisition of prokaryotic genes in fungi. Well, extensive horizontal gene transfer from bacteria to multicellular eukaryotes certainly did occur. That occurred, as you realize, in the evolution of plants and algae, when they acquired the cyanobacterium and the cymera that became the chloroplasts. That is beyond that. And this is and here we are talking about thousands of genes as demonstrated convincingly in this pioneering study of Big Martin and his collaborators. What about multicellular organisms, however, that do not have ancient stable endocymes? And here we decided about Unikonts. So to do this kind of to really, as we say in American, bite the bullet and do a very systematic, as careful as possible, analysis of horizontal gene transfer in the supergroup of eukaryotes, shown here as part of the sort of star-like tree of eukaryotes, that is known as Unikonts, which include metazoa animals, which include fungi, as well as amoebozoa and a few other objects. And so, here I will not discuss this in details at all, but just to say it very briefly, it took quite a bit of bioinformatic effort, so we developed this pipeline that applied a variety of tests to detect this horizontal gene transfer and eliminate all dubious cases by a combination of sequence similarity and phylogenetic analysis. And coming to the point, this is our current conclusion, which we are still validating to some extent, but which largely stand, I think. We have up to 1,000 of horizontal gene transfer events. Not genes, there are many more genes, but events where apparently a horizontal gene transfer occurred with possible subsequent duplication in the evolution of various branches of Unikonts. In this group, one candidate are well genes for which we simply do not detect any homologs in other in eukaryotes outside the Unikont supergroup. That is, we only detect homologs in Unikonts and prokaryotes. And group 2 are those where we do have homologs in other eukaryotes, but phylogenetic analysis indicates that horizontal transfer happens somewhere here in the evolution of this group. So I guess this is the key point. Here I show the fraction of the clusters of orthologous genes in different branches of Unikonts where we detect horizontal gene transfer, and this is on like horizontal gene transfer. This is an average more than 10% of gene of autologous gene classes. No small deal. Very extensive horizontal gene transfers. Considerably, it's the greatest in fungi, much less in animals, but still a substantial fraction of genes. We analyzed the different donors of horizontal gene transfer. I will go through this quickly. Just will note that these donors differ in different groups of Unikonts, and functions of these potentially transfer genes differ very much as well, but the generalization is that primarily these are enzymes, sugar and amino acid metabolism is most common. Many are actually poorly characterized enzymes where we just predict their activity and no more than this. These genes contain introns with intron density almost as high as on average in the respective genomes, but not quite. So apparently the accumulation of introns mostly has been completed, but to a small extent still is going on. And we have a variety of examples where we can show the phylogenetic evidence for grouping of these genes from animals and fungi, in particular bacterial homologs. I do not have the opportunity to talk about that really. So basically that is the conclusion, that a minority of genes, but a substantial minority, is involved, has experienced horizontal gene transfer. It's typical to see multiple transfers of the same genes. Mostly the transfer genes encode diverse metabolic enzymes. So this is what I want to finish with, basically. I just was proposing this, finishing this work very lately and did not have access to this most conceptual paper of the DRO with a very, very beautiful illustration, but I took an illustration that is almost as beautiful from a subsequent paper. It doesn't even matter that much what it shows, but what I want you to come away from this talk is this entanglement of different sources of various components of eukaryotic genus. Basically, this is one phrase that I think we should remember, that evolution of eukaryotes is a tangled rhizome throughout, from the beginning to this day. Thank you very much for your attention. This is my group in front of our building in Bethesda. Natalia Yutin, a senior postdoc in the group, was very instrumental in all these archaean reconstructions. Pere Puigbo was mostly responsible for the study of horizontal gene transfer, with the help of Sergei Mihedov. Yuri Wolf, who is hiding here, actually made the picture and I had to run it. Helped very much in the development of the algorithms behind these approaches. Bill Martin was my collaborator on a variety of conceptual issues as well as in some technical analysis. Miklos Churosh is a computer scientist whole from the University of Montreal, a lot in the reconstruction of the evolution of eukaryotic gene structure, and I think Didier is actually responsible for the most important ideas that I'm trying to discuss here. Thank you very much.
Well, I get one comment and then one other question. I'm very happy that I think that we discussed that yesterday, but I think that the idea is that the evolution has been parsimonious, is not reasonable. So there is plenty of different elements at different points. So there is scale steps, but there is plenty of change all the time. And I think that parsimonious analysis and trying to specifically for animals and biologists to get a single element is probably a mistake. it is what I think is interesting in trying to look at your hypothesis that makes sense for me. That of course eukaryotes could not be older than eukaryotes. That's it.
Prokaryotes could not be older than
eukaryotes. That's alpha proteobacteria, if you want.
Eukaryotes could be older.
Yeah,
right. That's excellent.
Yeah. So the idea of a branch of trees doesn't make sense at all. So there is something that's happened here. But if you look at the age of the alpha the speculation on the age of alpha proteobacteria, what is the branch of the arcade that is corresponding to this separation? Have you looked at that?
No, and this is a very, very difficult question. Extremely difficult question. We really do not know how old alpha proteobacteria are. We absolutely do know that before alpha proteobacteria emerged, there was a whole lot of bacterial evolution. A whole lot, evolution of proteobacteria, the double membrane, the blood, what not. A lot of differentiation. How long did it take in terms of time? It's very difficult for us to understand. Ditto about their key. We certainly know that a whole lot of diversification occurred early on. We do not have ways to date in any reliable way, so much to my regret, I cannot answer your question in any solid way. My speculation is that the diversification of both bacteria and archaea was very ancient. And actually, let me say something that is tangential already. Namely that what we actually tried to do, and others of course tried to do, was to date the origin of eukaryotes, or more precisely, determine the dates when the last eukaryotic common ancestor might have existed. I wouldn't get into the methodological details of this, but the conclusion that we come up with, the dates that we get with all the candidates are rather young, rather recent. That is, eukaryotes might have evolved between 1 billion years ago and 1.5 billion years ago. Incidentally, using completely different methods, and Philippe and others come up with similar conclusions. There is the sense that some other people think that eukaryotes are old. But if we accept the young lacquer, I would say there is little to no doubt that by that time, basically the entire diversity of both bacteria and archaea that we are aware of has already existed.
And this is a second question before it's a very nice idea to explain why the eukaryote gets a nucleus, but why do the first branch diverging of bacteria gets a nucleus?
You mean plantomacetes and the like?
Plantomacetes and Koonin
Okay. The plain answer is I don't know. I don't know why. But I would say it's not a nucleus. It's a compartment of writing that contains the DNA. That's for sure. 2 points, however. 2 clear points of distinction. First of all, there is a very specific set of proteins, a large set of proteins, dozens of proteins, that comprise the eukaryotic complex. We do not see